Okay, who are you and what do you do?
Yeah, so my name is Stephen Helms, and I'm a postdoctoral researcher. So that's what you are after you finish your PhD at the FOM Institute of Malft here in Amsterdam.
And what do you do there? What do you do during your every day?
I am studying biophysics, and I'm trying to understand how behavior works in a model organism.
And how behavior works? How difficult a question is that?
Yeah, so in general in biology there's this problem that there's a lot of molecules and genes and cells involved in everything you look at. And behavior is particularly the worst case because that's the output of what an entire organism does. So you have each cell, which has thousands of parts and things happening inside of it. And you have many cells working together, and you have neurons that cooperate together to make a brain, to make decisions and process information about the outside. And so it is definitely a difficult problem because you have to deal with all these different parts working together and figure out how, in the end, that makes something happen.
And why did you want to do the most difficult thing?
Yeah, so a lot of people in biology have gone from the other direction. So for the past 30 years or so, people have become very interested in trying to understand how particular molecules and particular genes play a role in different things. But I think there's been a lot of problems with this approach because there's so many parts. It's hard if you try to describe these processes in terms of all the different molecules, you end up with something very complicated. But in the end, we know that these systems, that biology does work in the end, and that it actually is able to evolve. And to be able to do this, there must in some way be a simple model that the organism is trying to accomplish, some simple description at a certain level. So I've decided that even though it seems harder, I wanted to start from the top and try to figure out what the simplest description we can have for what the organism is doing. And then from that, you can then try to fit in and explain, okay, this molecule is involved in this part. But if you start with all the molecules, you end up with something that is necessarily complex, and I don't think then you can actually understand the organism.
Okay, and behavioral, but you didn't start with human behavior. You started somewhere else.
Yeah. So I work with this model organism, a worm, and I actually have a video I can use to show you about this. All right. So this is from this,
is a microscopic roundworm, also known as nematode, that is 1 millimeter long. These nematodes are usually found in temperate soil environments worldwide. In the wild, they feed on the bacteria that help to decay decomposing plant matter. In the laboratory, however, C. elegans are typically fed lab strains of E. coli. C. elegans are transparent, thus facilitating the study of the cellular differentiation and developmental fate of each of its cells. There are two forms, hermaphrodites and males. Hermaphrodites have 959 somatic cells, while males have 1,031. The organism's neural circuit, made up of only 302 neurons, is extremely primitive, especially when compared to the approximately 100 billion neurons and the 60 trillion connections between them, known as synapses, that make up the average human brain. However, as a result of its simplicity, transparent nature, and the well-characterized cell lineages, the entire pattern of neuron connections, or connectome, of the C. elegans has been mapped. Despite the relatively simple neuronal system present within C. elegans, it displays numerous complex and primitive behaviors. Primitive behaviors include feeding, locomotion, and reproduction. Complex behaviors include learning, mating, or social behaviors. Researchers study behavior of these worms by observing how they are attracted to food and chemicals. Ultimately, behaviors reflect the activity of the nervous system. Despite their small size and simplicity, C. elegans are very useful to science, both to understand basic biological mechanisms and also to study basic mechanisms of disease that translate to understanding how to fight disease in humans.
The primitive worm. This explained it. So you and other people decided let's start with Well, relatively simple was being said, but relative in this case is very relative, I think, because it's still complex.
So that's the really interesting thing about this. So if you move to there are certain model organisms we use in biology. So the worm is one. The next more complex organism would be a fruit fly, which that has orders of magnitude more neurons. And then if we move to our brains, it's even far, far more.
Yeah.
So, but at the same time, many of the core features of what brains do, you can see in the worm's behavior. But because there's only 302 neurons, we have a hope of actually being able to understand it. And then there are many great experimental tools we can use. So we know all the cells of the worm, and they all have names. So I can go to a website and I can look up what a cell does and where it is. So I can show you that as well.
Yeah, show me.
So there's this great website called Worm Atlas that has information on the anatomy of the worm. So I can do things like pull up this AVA neuron. And so there's a left and right one, and you can see a picture of where the neuron is in the head of the worm.
Yeah.
And they have a list that says, what molecules it uses to communicate with other neurons, and what sensors it has, and then the known functions of it. Mm-hmm. So this neuron, for example, is an interneuron. So it's a neuron that gathers information from other neurons and makes decisions, and it's involved in controlling the motion of the worm. So we have all these great resources like this, and we can also experimentally manipulate the worm. So if I want, I can go in and I can use a laser to kill this particular neuron and see what happens to the worm. So that's how they do things like this and figure out that it's involved in this case, making the worm go in reverse.
Yeah. And is this a sort of a Wikipedia sort of thing? Do you put stuff in it yourself as well? Is your material in here as well?
Yeah, so this one is not like Wikipedia, but there's another community resource called Wormbook, which is an open source textbook people write for the worm. So that one I could, for example, write an article on the motility of the worm and put it on there.
Yeah. And let's start, or let's pick this issue.
Yeah.
How different is it, say, working together with colleagues in your field now than it used to be? Because this must be ideal compared to what people used to do a long time ago.
Well, I think this is different than some other fields, but it's actually been part of the nature of this worm community since the beginning.
Yeah.
So it's quite strange. Sometimes in science, communities can have different personalities. So the worm community started in the mid-'70s there's previous research on this organism, but as a model for understanding the nervous system, it was really started by the Nobel Prize winner Sidney Brenner in the 70s. Mm-hmm. And so he picked this as a good model for understanding how a brain works. And the community is still relatively small. So for example, this year I went to the International Worm Meeting where most of the researchers go, and it had, I think it was 3,000 or 5,000 people. So it's big, but at the same time, everyone knows each other and they all work together and try to make it easy to pool information to understand how the simple organism works.
Yeah.
But yeah, other fields, sometimes people can be very competitive or they don't easily share things.
Not sharing wise, but this is
But this is like a sort of like the open source community in like programming. This is sort of the equivalent in biology.
Okay, nice.
Yeah.
And how far are, let's say, the 5,000 of you in understanding this worm? Yeah.
So the community started off with a focus on genetics, and there's been a lot of great work done in again building up these resources. So, for example, we have tools for manipulating the worm genetically and for identifying the genes involved in different behaviors. But I still think, despite all the information we have about the parts, we don't have a very good understanding of how the nervous system actually generates behavior. And I think part of it's a problem of not having a good definition of what the behaviors are. So because it started off as a field focused on genetics, people picked, when you do genetics, you look for things that you can easily, you try to basically break something and then see a big effect that you can visually see. Because you have to basically find, if you have some plate where you have a lot of worms and you're looking for one that is not behaving normally, you want to see something really obvious.
Yeah.
So they have things like, one of the phenotypes is called uncoordinated, which means that the worm can't coordinate its movement when it's trying to go. So you see worms that either can't move at all, or they go in circles, things like that. But these aren't good quantitative. We can be more quantitative with how we describe what a worm is doing. So that's what I'm trying to do in my research, trying to find the best possible, but still intuitive description we can have of the behavior.
How do you do that research?
Yeah, so what I do is still very much the classical experiments. I put worms onto a plate, so just a dish that has some a gel on it that the worms can crawl on top of, and then I track their movement while they're exploring that environment. So I can show you a picture of that. Yeah, so there's a picture here from my experimental setup of a plate, and there are 4 worms placed within these 10 by 10 millimeter boxes.
Yeah.
And then I track them for 30 minutes. So I just record a video, and then using a computer algorithm, I automatically find and characterize where the worm is. So I can measure things like the shape of its body, where its head is, and where it's located. And if you follow that for 30 minutes, you get something that looks like this. So these are 2 different species of worms.
Mm-hmm.
And the box here corresponds to this 10 by 10 millimeter grid. So you can see, like, on the left is a worm where it explored most of the space available to it.
Yeah.
And on the right is a case where the worm stayed within a very small spot.
Yeah.
And these actually turn out to be very biologically relevant things because these are different motility strategies that worms take.
So, and for example, what does this learn you, or how does this help you?
Yeah. So what people normally would have done is they would simply look at the overall thing and say, okay, this one explored a lot and this one explored very little. But what I do is I take this data. So this is just one snapshot at the end of the experiment showing you where it went. But I have data for the movement of the worm over this entire period. And you get a lot of data points that characterize its movement. And it's a great system because you can get a lot of quantitative information out of that. So I'm building models that capture all the intricate features of what the worm is doing, and then trying to use this characterization to describe the behavior.
Yeah. Why is it important that we understand all this?
Yeah. So again, there's this general problem that we don't understand how the structure of the nervous system, which in the case of the worm, we know exactly what the structure is. We can't directly say, okay, this worm is moving a lot because there's something special about this neuron. Mm-hmm. People try to do this and they get sort of fuzzy answers. So they find out, yeah, if you look under one condition, you can see that if you kill this neuron, then it's somewhat affected, but not completely gone. Or you can see that if the worm, for example, smells something, you see some neurons activate. Because we have these great tools where we can watch visually the activity of the neurons using a certain protein that glows when the neuron activates. Mm-hmm. But it's been hard to say basically that this neuron does this thing or is even completely required for this thing. It seems that, yeah, it's always some very complicated answer. And I think that it's partly a problem just because the way in which we're trying to look for this connection, so we're trying to say this neuron is doing something, but if our description of what we're looking for it to do is not quite optimal, then it's hard to get clear answers. So I'm trying to find mathematically what the correct way to describe what a worm is doing is. Mm-hmm. And I think this is the key then to actually go back and say, okay, now it's really clear when these neurons have this pattern of activity, then you see this behavior happening. But if you try to do it without clearly having a good definition of what you're looking for, then you may be that you're mixing together 2 different things, or you're just not quite capturing the richness of what it's doing.
No. I say there is the phase comes that we you understand the worm and its behavior, and then you've been to the fruit flies, and then to the humans.
Yeah.
Say if you would understand how my brain works, what would it help us?
Oh, yeah. So, yeah, again, this is a general problem in biology, but right now, a lot of the things like the medicines we take, and particularly the case for like antidepressants and things like that, there's really no rational basis for them. They are found kind of randomly. Yeah. They tested chemicals and they found that some of them made people happier. And we don't really understand how these things work. And so most drugs that are designed nowadays are just variants of these originally discovered things that were kind of randomly chosen. And in some other fields, not in the neuroscience field, people are trying to rationally design new drugs, but often it fails because they basically found some protein in the cell that's important for something like you're controlling your blood pressure, for example. But then it turns out that when you study it in the context of a whole body, it doesn't end up working. And I think it's, again, this problem that we don't have a good understanding of what is actually going on inside of organisms. And this is particularly the case for our brain. So I'm hoping that by developing tools here for theoretically understanding how the nervous system works, then when we look at more complicated things, we have at least some hope of rationally understanding what is going on.
Yeah.
Again, right now it's not so much a rational process. I mean, there's an effort to do this. I shouldn't dismiss what people are trying to do, but it's been a huge problem.
But it's such a long-term work what you are doing, of course. So what makes you, when you come home, what makes you think, well, this was a good day?
Oh, yeah. So I think it's a good day when I find some simple explanation for something complicated that happened. Yeah. So sometimes it's like, you have data that looks really weird and complicated, but if I can find some simple theory or I can look at it in some nice way, then I feel content. I'm really trying to find, I believe fundamentally that biology is not complex, that there is some simple rules underlying it, and I'm trying to figure out what those rules are. So any day that I find some simpler way of describing something that I've been looking at, I'm happy.
Yeah. How does computing help you in your work?
Yeah, so as I was mentioning, there's a lot of data here. And the key to this model building approach is to really amass as much data as possible, because I really want to take the raw data and use that to directly build models. So right now I'm kind of operating on the limits of what I can do computationally. So I've worked out experiments where everything is automated up until the point where I'm actually, thinking about the data myself. That I can't automate. But so the amount of data I can process right now is being limited by the fact that, for example, my desktop computer, it takes 3 times as long to process the data as it takes to collect an experiment. So I can keep doing lots of experiments, but I end up with this huge backlog of data to process. And then the storage is also a problem. So again, I track these things by using video microscopy. And these videos are huge. So the raw video file is 240 gigabytes per hour.
Okay.
And there are many, many experiments. Yeah. So storage becomes an issue as well.
And how are you? And what are the solutions to these issues? Because they always say storage is getting cheaper every day, and
storage is getting cheaper. But then we're also the amount of data we can collect is quickly growing as well. So, for example, I have been looking into how I can possibly upgrade all the things I'm doing at the moment. And I could potentially scale up what I'm doing and make video files that are something like probably 10 times bigger. So I would get more data at once, but I would be collecting even 10 times more data. So that growth rate is still on the same order of how much the storage cost is going down. So I can buy multiple terabytes of hard drive space, but on my own it's difficult. So I'm thankfully here in the Netherlands we have actually very good resources. So the SurfNet and SurfStar organizations have amazing computational and storage space, and so they're very helpful at dealing with these sorts of things. Yeah,
can you tell me something about? Because how does that go? Do you do you have to make a reservation for storage, or a reservation for can I use all your computing power next week? Or
so I'm still learning myself how to do these things, because I've only actually been working here for about a year and a half, and I'm just not getting to the point where I really need to do these things. But in general, the different organizations here, for example, my research institute has an agreement that we can use something like, so many thousands of computer hours per year or something. So if you're within this limit, you can do certain things. But these agreements right now are mostly for this kind of still older technology. So they're newer things like I don't know if you've heard of cloud computing. It's this idea that you have many different computers that are all linked together, and you can just basically say, okay, I need to have this much power, this much space right now, and you can adjust basically the amount of power you have.
Couldn't you do everything with Amazon services?
So, and that's an alternative thing, but I mean, they're not designed for doing academic research, and they have different pricing structures. So it's better to use a kind of local thing with actual people here who can help you out.
Yeah, but you do feel that, say, computing power and pricing, etc., of storage is hindering your. Would be helped with better facilities over there.
So I certainly see that. I can already start to see interesting things with the data I have, but I really would like to have about at least 10 times more data than I have, and to do that, I would really start having troubles with the amount of power and storage I have available to me. So right now I'm already using up basically half of my lab's storage space for my one experiment I've done so far. Yeah.
So how do your colleagues look at you?
Yeah, well, my colleagues are also generating lots of data, but the computing support people at the university, they're not so happy. They keep thinking, why do you need all this data?
And is it, like you say, because is video the thing to do, or couldn't you do it with sensors or stuff, so that it's less data intensive? Yeah,
there's always ways you can try to reduce the data, but in general in science, the philosophy is you want to try to keep as much of the raw data as possible, because it may turn out that later on we thought we only need to measure one thing, but there was actually something else interesting that was going on.
Yeah.
So I do at least compress my data and then get rid of the uncompressed video, because for what I'm doing, in the end, you process the image so you just see like an outline of a worm. Mm-hmm. So I don't need to know so much about the rest of the video. But I do want to see what the worm looked like and where it was. It might be that there's something weird in my experiment, and when it's in a certain place, something weird happened. Yeah. And so I want to be able to visually go back and see what was going on.
And can you tell me something about how you work? Because of course you tell about what you are doing. Is this a team of people working at this?
So right now I'm the only one doing the actual experiments, but I have a team of people who help me analyze and think about the data. So as a postdoc, I still have an advisor at my institute, and so he oversees the experiments and gives me a lot of advice about what I should do, on a weekly basis. And then I have another collaborator who is a theoretical physicist, and he's at the Vrije Universiteit here in Amsterdam. Yeah. And he's also in Japan for 20% of his time. And he helps me out a lot about thinking about how to characterize the data, how to build these models. And then I have another collaborator in the US who is a neurobiologist, and he specializes in more this level of the neurons and the genes involved in connecting the physiology of the worm. So for example, when worms are hungry, they behave very differently than when they have eaten a big meal recently.
Are they more busy, or what do they do when they're hungry?
So there's a kind of funny thing you can do. You can starve them, and then if you give them a big meal, then they go to sleep. Sort of like you and I, if we have a nice feast at the end of the day.
Yeah. Is the world of computing develops really fast? What does that mean for your work? Because say a couple of decades ago, a lot of things seemed relatively static. And you could spend a lot of time making maybe things better. But now you're living in a world where everything changes as well. So the computing power, the cloud computing, etc. So what does it mean for your work?
Well, I find it very exciting. So before I went into science, I was also thinking about going into computing because I always thought computers were fascinating. And then I later figured out that biology is an even better computer than the computers we build. So I think for me, it's fun keeping up with the new technology, but it also is something. It's important to constantly pay attention to the new things you can do, because it's opening up a lot of new possibilities now, because we can collect so much more data. We can analyze it in ways we couldn't do years ago because we didn't have the computational power to do it at a reasonable time scale. Yeah. So it's exciting that we have all these new opportunities, but it definitely requires that you keep up to date and talk to people who know how to do these things. So I've been learning a lot from people from Surf, for example, about new computing technologies that I sort of had vaguely heard about but hadn't previously read any information on, and how they can help me in my research.
Does this mean something for universities? Do you need to school people more for change than for an end set of skills?
Yeah, so I think, things like programming is very important. You have to have some level of computational skill in science, but it's more a matter of figuring out, what things are available to you. So a lot of times it's easier just to use your desktop computer because it's in front of you. You can easily see it, interact with. Some of these things like cloud computing, you can't go and look at the cloud computer itself. So it's hard to sometimes know I can do this thing with this, or this thing's available to me. So it's important, I think, that at least the support people at the local institutes, they know about these things, and then they can at least tell you, maybe you should look into doing this thing or that thing.
When do you think that you and your colleagues understand the worm?
Yeah, so for me, I would just be happy if I could say, if I see some changes in behavior, and I can say how the changes in some neurons I can look at in the worm, if I can explain everything about how the worm's behavior changed by explaining the activity of these neurons. And I think that's something that's achievable over a 10-year time scale. Yeah. But then the problem is going to be scaling it up to more complex organisms.
Yeah.
But if we can't do it for a worm, it's going to be very hard to do for a human.
Yeah.
But there's still something that both...
But isn't it fascinating how I find it so fascinating. Everything we do for ourselves is so natural, and that it's such a complex thing that we can't even yet understand ourselves.
Yeah. It's, people sometimes say if the mind is... I don't know where this quote comes from, but it's some famous quote like, if the mind were so simple that we could understand it, then we wouldn't actually be able to do that. Yeah, it needs to be complex to some degree to do this. But I think the key point is that there is something simple in biology that we can understand, because if we look at evolution, we know that evolution works and you can change, for example, behaviors dramatically. We don't behave exactly the same as an ape or as a mouse. Mm-hmm. So somehow these things can change in order to do different things on reasonable timescales. And if it's something really complicated, that's an impossible problem. If I give you this room full of random switches and you have to find some combination of them that makes the light come on, then you're never going to solve it. You have to have some sort of simple structure there.
Yeah. How do you said the community of people busy with the worms is an open community, is a collaborative community. So how does it go? This go? Is there a sort of a site, a news group, whatever it is, where you meet each other every day, every week?
Yeah, so then there are meetings that happen every year. That's kind of still the main way in science that we communicate with each other. But then there are also forums, and for example, my collaborators, I talk to them regularly. So it's kind of, the people I work with most closely, I talk to more on a weekly to monthly scale, and then the broader community is more on a 6-month to 1 year scale at different regional meetings, or if I have some problem. Like for example, I have a new student who started a few weeks ago, and we're waiting on some reagent to come in, this certain worm that had a molecule that makes its neurons glow. And it didn't arrive on when it should have. So then I sent an email to all the other worm researchers in the Netherlands, and then one of them at Erasmus sent me an email back, I have lots of things you can use. So then I sent my student down there to go pick up some reagents.
How many are there in Netherlands, do you think?
So the number of professors is probably something on the order of 10 to 20. So when we have the Dutch worm meeting, which has the students plus the faculty, it's something like 100 people.
But it must be brilliant to be at such a day where you are all studying the same thing.
Yeah, it's very nice because you can get together and then you can hear what other people have to say about it, and you learn all the latest tricks about how you can do things.
Yeah, and so very often you have to tell it now like you're telling it me. So you have to make everything really simple. It must be so much fun that you can talk at the same level.
Well, but even other researchers, people often think this is the case in science, that because I do science and so the person does science, I can then immediately talk about these complicated things. But even in my own institute, most of the people don't work on neuroscience, and so if I start talking about a lot of jargon about how neurons work, they're not going to necessarily know either. So it's always it's useful actually to try to break it down into some very simple description, because if you can't do that, don't actually understand it.
Yeah, because like you said, that is your most important goal. Make it understandable.
Yeah, exactly. Yeah.
Okay, thanks a lot.
Thank you.
Bye.